Well, generic answers can be very economical. Now we move from the regulatory scene, the overview, to 3 case studies. And the first case study will be presented by Stefan Mühlebach, who is Chief Scientific Officer from Vifor. And please, Stefan, take the floor. He's also chairing the NBCD working group.
Thank you, Daan, for the introduction, and thank you to having the opportunity to speak here on this industrial case related to iron nanoparticles. And I will focus on those products who are made to give iron, let's say, to treat iron deficiency. You have here my appointments in the sense of giving transparency as well. Why treating iron deficiency by this kind of nanoparticles? Just to briefly show you the medical need we have in that area. So the iron homeostasis is very well regulated, and iron deficiency can pop up if there is an increased loss of iron or an increased demand. The most important carriers of the irons are carrying iron 3. That is, on the one hand side, the ferritin, which is in the tissues and gives you the storage. On the other hand, it's the transferrin. Now, what is the medical need or the problems we have with that? Looking into a small physicochemical aspect, it would be nice to give IV iron 3 as an infusion, but that is not possible because of the physicochemical characteristics. So iron 3 will, under physiological conditions, immediately precipitate. So there is an only way to go for that if you have polymeric iron oxyhydroxy cores, which are combined with carbohydrates as a ligand, which stabilizes, on the one hand side, the polymer core, and on the other hand, keeps these kind of colloidal particles in solution. If we take a cartoon of iron sucrose, which is probably the mostly used IV iron since decades, and where there is a lot of experience in handling this drug, then you can have a look here and see how this product is made. So we have, as said before, the central polynuclear mineral iron core. Then we have, because of this specific manufacturing, different sizes in the core, but as well on the whole particle, which is mainly defined by the carbohydrate, its chemistry, and finally the nanoparticle characteristics. So PK, and through that PD and safety and even immunogenicity is mainly concerned or influenced. The stability of these products are very much depending on the specific conditions. So, for example, looking again to iron sucrose, the stability in vitro, meaning in the vial, is completely different than the one if you have injected the drug, just knowing that the pH in the solution and the colloidal solution is in between 10 and 11, whereas physiological conditions are completely different in the area of 7.4, which destabilizes quickly these kind of products. Looking in these colloidal IV iron carbohydrate product, there is quite plenty of different products which show differences, mainly, as I said before, by the ligands used. And taking again the example which I have announced in the beginning, and looking into the specifics, you can see that one of the aspects to characterize such drugs is the ligand in itself. It's sucrose in this case, that's a disaccharide, but there are other ones like monosaccharides, there are branched polysaccharides, etc., which have an immense influence on the size of the product, but as well on the kinetics, on the half-life. So looking into that, there is clear that if you have different composition, there are differences. But it's more interesting to know that even within a group of these NBCD products, there could be differences looking into batch-to-batch consistency, but as well if we have follow-on versions. So as we said in the beginning, the manufacturing is key to give the characteristics to this product. So the complex processing, the different controls, the starting materials are highly important to get a comparable product by the end. And by this manufacturing process, on the one hand side, the pharmaceutical equivalence is defined, which go through the specific nanoparticle formulation, which, as we said in the beginning, is not something you can fully characterize with given physicochemical methods. And the structure surface morphology of these particles is highly important for the interactions with the biosystem. The kinetics are influenced, and the pharmacokinetics as a follow-up. So the in vivo performance mainly depends again on these manufacturing aspects. When it comes to follow-on versions, the question is, what are the important aspects to check for, or how much similar such a product has to be? And as we said in the beginning, as we have no full picture about the composition of such a product, it's not easy to give the necessary indications. So on the one hand side, some characteristics about that. It's a synthetic procedure, which is different to the biologics. We have so-called non-homomolecular structures, polymeric ones. The physicochemical characterization gives not the full picture, and there might be unknown structural element that can impact kinetics, but as well therapeutic performance. So what are the clinical meaningful differences, or let's say the summary of evidence, to give comparability in between a given product which is developed to a reference product? And the manufacturing process is critical, very much complex, and often intellectual property not disclosed outside. What do we have as experiences out of it? And I will probably go through some published data which were done when comparing an iron sucrose, the originator, with so-called iron sucrose similars, follow-on versions. It is from a publication by Tobli et al., who was using different ISSs and iron sucrose in an animal model to look on how the drug was distributed and finally popped up when looking into these animals. He gave 40 milligram IV iron, which is an unphysiological high dose, but nevertheless, the idea was not to give a picture, principally to give some indication on the human situation, but just to look into these different products. And what popped up were this kind of histology. You see on your right hand on the lower part the control, which is a saline solution. You see up there the Vifor, which is the originator product. And you see on the left-hand side two ISSs, different ISSs. And even if looking into this Prussian blue staining, you can see that there are differences. And these differences, when tried to analyze statistically, clearly showed that on the one hand side, there is a difference in between the control and all the treated animals. But again, there are significant differences in between the originator and the test ISSs. And the picture indicates, among others, that there could be a reason why, for example, oxidative stress could be different. And this was correlated as well with other determinations of enzymes, of redox aspect, even going down up into organ functions. So there are significant different deposits in the ISS tests, which could be, as I said before, an indication why some of these products behave differently in animals, but most probably as well in patients. Another paper which came out was related to an authorized ISS in France, where in a population of stable hemodialysis patients, there was a switch on the product during treatment of their iron deficiency. So in the left-hand side, you see the treatment with the target of the hemoglobin, which was achieved in a rather stable dosing. Then there was a transition period, and after this transition where the product has been changed without knowledge of the physician that happened, an immediate drop of the hemoglobin occurred. And to finally then re-establish this situation like in the situation ahead, it took a long time to put the patient back in their stable aspects, and that was accompanied by a major increase in the iron dose you needed and in the darbepoetin, the EPO dosing. So on the one hand side, what you intended to have by follow-on versions, meaning that some economical benefit could result, the opposite occurred. And the second aspect is, where did this iron go? Because normally mankind are not able to excrete iron, and it's somewhere in the body. And that was one of the important issues. About a third of the dose of iron had to be increased, and about 13% of the ESA dose just to get similar results. So what can we learn out of these aspects, and how can that add to future development and probably harmonized regulations in this assessment of such nanocolloids or even the iron nanocolloids? On the one hand side, these products are complex. They have a three-dimensional structure, nanoparticle structure, which is important for having functions and be taken up and handled by the body. The size, the morphology, and the surface affects the PK. So, for example, we know that the uptake in the body is taken by the innate system, and this innate immune system is having a phagocytosis in that active aspect. In addition, these iron products are products. So the release of the active ingredient is given within the body through a well-regulated internal system involving even active and passive transport mechanisms. And this interacting with this immune system is a key element of the whole process, but we have not yet good models to reflect that and to check for that. And as we have to deal with an endogenous component, it is very difficult to do good studies in that, indicating the bioavailability out of these nano products, on the bioequivalence, on the area under the curve, and we have probably to correlate kinetical aspect with biomarkers, and then to look in different dose ranges, because anemic patients are different to those who have a normalized iron content, and all of that has an impact on the product. So a stepwise approach for all these products makes sense, going from quality to non-clinical and clinical, probably head-to-head analysis. And finally, the therapeutic equivalence is the big challenge. To what extent we are able to interchange, substitute products, and how can we give this rationale that this drug can be used in a similar way. With that, I'm at the end. Thank you very much.
Thank you very much.